Hello, everyone, and welcome to today's live broadcast, Promise and Challenges of iPS Modeling in Parkinson's Disease. I am Soon Pham of LabRoots, and I'll be your moderator for today's session. Today's educational web seminar is presented by LabRoots, the leading social scientific networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you today by Cellular Dynamics. Cellular Dynamics International is a leading developer of fully functional human cells derived from induced pluripotent stem cells, or iPS cells. Our iCell and MyCell products provide industrial quantities of high quality, highly pure human cells enabling disease modeling, drug discovery, toxicity testing, and cell therapy research. For more information, please visit www.cellulardynamics.com. Now let's get started. You can pose questions anytime during the presentation, while they're fresh in your mind. To do so, simply type them in the green Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you have problems viewing or seeing the presentation, just click on the support button that's found at the top right of the presentation window, or type them in the green Q&A button on the lower left corner. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left corner of your webpage and follow the process of obtaining your credits. I would like now to present today's speaker, Dr. Birgit Schuhl. Dr. Birgit Schuhl obtained her medicine and her doctoral degrees at the George August University in Göttingen and Medical University in Lübeck, Germany. Dr. Schuhl worked during her neurological internship at the Medical University Lübeck with Dr. Christine Klein on genetic forms of Parkinson's disease and dystonia. She also completed a postdoctoral fellowship in the laboratory of Dr. Uda Franke, Department of Genetics at Stanford University School of Medicine, before joining the Parkinson's Institute in October 2005. Dr. Schuhl leads the genetic and biomarker program since 2008. She has built a stem cell program developing novel disease-in-a-dish model of Parkinson's disease using patient-derived induced pluripotent stem cells. In 2014, she became the director of the Brain Bank at the Parkinson's Institute. I will now turn it over to Dr. Birgit Schuhl for her presentation.
Thank you so much, Sue, for the introduction. Thanks for CDI for sponsoring this lecture, and I'm really excited to give this presentation. I know my lab is joining me also, and they're sitting next door in a conference room, so let's get started. For those of you who don't know the Parkinson's Institute, we are in the San Francisco Bay Area. The institute was founded in 1988 by Dr. James William Langston, also called Bill Langston, and the exciting part of the institute is that we have a clinical center who has about 1,500 active patients, a basic research laboratory, and clinical research all under one roof. So it's very easy to conduct translational studies. So that's why we have large tissue and biospecimen resources like the brain donation program, the neurogenetics program, or also the stem cell program, which we started to establish in 2008. So let me start with what is Parkinson's disease. The textbook definition of Parkinson's disease is that it's a slowly progressive neurodegenerative disorder characterized by motor symptoms of rigor, tremor, and bradykinesia. It affects about 1 to 2% of the population over the age of 60. And pathologically, we find a loss of dopaminergic neurons in the brainstem, characteristic protein inclusions known as Lewy bodies, and those Lewy bodies contain a protein that's called alpha-synuclein. And research has been done over the last almost 20 years to figure out what alpha-synuclein does. You find it, as I said, here in Lewy bodies, and this is an alpha-synuclein stain in 1997, soon after the gene was discovered as the first causative gene for familial Parkinson's disease. So again, the clinical aspects are resting tremor, bradykinesia, stiffness, postural instability and a phenomenon called freezing, so patients can't initiate the next step. For example, they can't walk through a doorway or so. Down here you see a panel how the disease is progressing over the years. Usually patients come within a year to a neurologist and get the diagnosis. When the medication is working fine, there's a 3 to 5 year honeymoon period where the patients do pretty well. That's followed by a motor complication period where the medication is not working so well and you have a lot of side effects, for example, dyskinesias. At some point, the symptoms become resistant to medication, and then in late stages, there's also a cognitive decline period. So some of you might have heard earlier today Dr. Carlson from Sweden, but just to give you some history about the dopamine deficiency in Parkinsonism. So Dr. Carlson did some really seminal experiments, and he found out that levodopa reversed bradykinesia in mice and also rabbits that he treated with reserpine. And he speculated that dopamine deficiency causes Parkinson's disease. He proposed that, but early on people didn't quite understand because they thought that dopamine is just a precursor and not a neurotransmitter in the brain. But he was proved to be right, and he won the Nobel Prize in 2000. This is just one of the pictures from his publication where he had reserpine-treated animals, and then after they were treated with dopa, the symptoms reversed. That's quite exciting. That was picked up pretty quickly by Ole Hornykiewicz, who then showed that in the striatum, in the brainstem of Parkinson's patients, there's actually a depletion of dopamine. And these early doctors, Barbeau, Bergmeyer, Hornykiewicz, and Kotzias, they treated soon after patients with L-DOPA and saw a dramatic effect. And then Jahr conducted the first clinical trial, and the effects were so dramatic, despite the dyskinesias that they already noted at that time, that L-DOPA received regulatory approval and became really the main treatment for Parkinson's disease, even nowadays. Are we missing something in this picture? This is a case history from a patient that Dr. Langston saw about 10 years ago, and it is a 70-year-old man presented with a 15-year history of chronic constipation. 10 years prior, he lost his sense of smell. He also developed an unusual sleep disorder, and he was hitting his wife because he had so vivid dreams, and he had several episodes of depression in the past. So none of the typical clinical motor symptoms were presenting, but these are symptoms that are also associated with Parkinson's disease, and they are called pre- or non-motor symptoms of the disease. Pre, because they can occur before the motor symptoms arise, and, as I said, non-motor because they're not affecting movements. So it's olfactory dysfunction, depression, and the sleep problem he had was REM disorder. And then besides the constipation, there could be also other autonomic disturbances like reduced cardiac innervation, hypertension, urinary or erectile dysfunction, constipation, and fatigue. And as I said earlier, in later stages, also cognitive function declines. So there are correlates in the brain that you can attribute these for. So for example, the olfactory dysfunction, you can see the olfactory bulb is affected. For depression, the raphe nuclei and other areas in the brain. For REM disorder, the nucleus ceruleus, autonomic disturbances, you attribute that to changes in the sympathetic ganglia, dementia, weight loss, and hallucination. So as I can show you, it's not just the substantia nigra that's affected, but also various other brain areas. So, and just to tell you that this is not something that has just recently become apparent already. Heinrich Lewy, back then in 1912, reported Lewy bodies not in the substantia nigra, but in these other brain areas. For example, dorsal motor nucleus of vagus, nucleus basalis minor, and hypothalamus. He's not reporting the substantia nigra. And then in 1928, another German pathologist, Herzog, reported Lewy bodies in the sympathetic ganglia. So we know about this phenomenon all the way since its beginnings, but I guess the dopamine discovery was so profound that we might have forgotten those early discoveries. Later then, Kwarman found Lewy bodies in the esophagus in 1984. 1988, Wakabayashi reported Lewy bodies in the lower intestinal tract. So already very solid information that there's Lewy pathology not only in the substantia nigra. This is a slide from Professor Heiko Brack in Frankfurt, and he saw Lewy body pathology and Lewy neurites here in the stomach of patients. Okay, so I think what I convinced you about is that Parkinson's not only affects the substantia nigra, there's also prominent neuronal loss in other areas like the noradrenergic locus coeruleus, locus basalis of Meinert, Ralphi nucleus. Those are not dopaminergic systems, and Lewy bodies have been found in the same areas. So now I think we figured a lot of things out clinically and pathologically. However, there is, from a study in 2014 from the Arizona Study of Aging and Neurodegenerative Order, a paper that says there's really low clinical diagnostic accuracy of Parkinson's disease, and it's quite devastating. You can see the numbers here, only 20%, 26% accuracy for clinical diagnosis of Parkinson's in untreated or not clearly levodopa responsive subjects. The diagnostics get a little better when people are responsive to medication. It rises to 53%, and then I think clinicians get pretty good when there's a longer disease duration. When the patients respond well to levodopa, then there is a greater than 85% diagnostic accuracy. So in summary, what we can say is Parkinson's disease is a complex disorder, and not as the textbook definition in the beginning, it's confined to the substantia nigra, but to many different areas. So we'd like to call the Parkinson's disease now multi-system Lewy body disease. This is from a review that we presented late last year in Nature Genetics. So is Parkinson's genetics? That's the question that I get a lot of times from patients. And what we see in the clinic that about 10% of patients with Parkinson's disease report a family history. And since the discovery of synuclein, alpha-synuclein in 1997, there have been now over 30 genes or risk factor that have been attributed to Parkinson's disease. So either by linkage studies in families or exome sequencing, or by genome-wide association studies. The concern here is that maybe not all of those genes actually cause Parkinson's disease, rather than a form of clinical Parkinsonism, because some of those loci that are described here really don't show any Lewy body pathology. The genes that I've highlighted, SNCA, so alpha-synuclein and LRRK2, and also on this side, GBA, are the three genes that are most common and really result in a syndrome that is really closely related to multi-system Parkinson's disease. So to look at this a little closer and also to visualize that, because in a table things become more equal, what I was trying to do is to draw a table and a graph, and I wanted to illustrate how close are the other genetic forms to idiopathic Parkinson's disease. So Parkinson's disease is represented here. On the x-axis, you see what we call clinical distance, so it's a Euclidean distance of different clinical symptoms. And on the y-axis, I was plotting the age at onset. The different colors represent the Lewy body pathology. So blue is typical Lewy body pathology, and then yellow and orange are ones where there's either no information about Lewy body pathology or really no Lewy body pathology found. The size of the circle really represents the prevalence. This is just relative because Parkinson's disease sporadic forms are over 90%, so we use the logarithmic scale here. So what you can see then is that really only LRRK2, the GBA gene, and alpha-synuclein are the ones that come closest clinically and neuropathologically to idiopathic Parkinson's disease. So in summary, what I wanted to convey is that there are many areas in the central and peripheral nervous system that are affected. Besides the dopaminergic system, there's also the noradrenergic, cholinergic, and serotonergic system affected. Genetics gives us a big opportunity to understand the disease, but the challenge is also that we might lump some of the syndromes together that should be subclassified. And we believe that only 3 genes to date, and more discoveries coming along the way, I hope, and only 3 genes, LRRK2, GBA, and alpha-synuclein, really cause a form that we call now multi-system Lewy body disease. So now I'm coming to modeling Parkinson's disease, and what is the right model to find disease mechanisms, to use the cells possibly for drug discovery. So I was listing here all the human-derived cellular models that are available and where people did great work with. So you can start with primary patient-derived cell lines like fibroblast, EBV-transformed lymphoblastoid cell lines, cygoid cell lines. Then there is the category of human neuroblastoma or other carcinoma cells. An interesting model are also immortalized human mesencephalic cell lines, and one of the most famous one is the Loomis line. And then now there are stem cells that we can use for modeling disease, either in a dish, or we can use them for cell replacement, or also drug discovery. So this just illustrates how we use a human model, and we're using induced pluripotent stem cells from patients with Parkinson's disease. And it's a cycle that works really well. So you get in the clinic, get a biopsy, use an explant culture to bang some of those fibroblasts, and then use a technology called epigenetic reprogramming, and you will receive pluripotent stem cells in the dish. To model Parkinson's, you want then to differentiate those cells into neurons, and then you have really a wide variety of what you can do with those cells. Disease research, drug screening, genetics, or developmental biology. The other approach that people also use is to try to use pluripotent stem cells or similar stem cell types for cell replacement. So this is how you can envision modeling Parkinson's in a dish, taking a biopsy from a patient, use them to reprogram them with several factors, and then do direct neuronal differentiation to study disease mechanisms, or, as we say, we can conduct clinical trials in a dish for drug discovery. So since 2008-2009, we derived over 80 skin cell lines here at the Institute. We have over 100 induced pluripotent stem cell lines from those. And in this table, you see the different genetic forms of the disease. We have alpha-synuclein, and you see LARC2, in which we have really most of the cases. And then we have some rare forms of ataxin or 22q11 deletion. I also want to add that we recently started a program where we were now collecting meninges cells, which could also be another way to really look at iPS cells that come from pathologically confirmed Parkinson's disease. So this just is a nice overview and a teaser of what you can all do with this type of research. And I wanted to show you that we used a patient who is descending from the Iowa kindred that has been followed at the Mayo Clinic for over 100 years. He has a very strong family history in his family, and it's a very early onset. So the typical onset is not in the 30s in Parkinson's disease, as I said earlier, in the over 60. So pathologically, we have Lewy body pathology, and this is really all over the brain, including the cortex. So in this case, what the group around Singleton found in 2003, this is a family that has a triplication of the alpha-synuclein locus, and that is depicted here. This is a copy number analysis where you see actually 3 copies of that gene over a range of 1.7 megabases, and alpha-synuclein is here included. So in fact, what you have in your cells are 4 copies of alpha-synuclein, 3 copies from, in this case, from the maternal lineage, and 1 copy from the paternal lineage. And I extended on the right side here a pedigree, and the patient that we're actually following is this one down here, who has Parkinson's disease, and his disease started at age 37 or 38. So over the years, we were able to derive fibroblasts. We created iPS lines. We studied neuroprecursor lines from these iPS cells, and then we looked at cellular phenotypes, and we found profound mitochondrial phenotype and increased susceptibility to oxidative stress. And this has been published in 2011 and 2014. So I want to talk a little bit about the challenges of iPS modeling. Since you're working with human tissue, you have multiple donors. You create in the dish multiple iPS clones, and you create also, when you're differentiating different types of neurons, and you can use different differentiation methods. So I want to illustrate what are the things you should consider when you're working with iPS lines and where the challenges could be. So one is the background of the tissue donor. So number one, what's the source of donor tissue? Age, gender, ethnicity, the disease that the subject is presenting with, the underlying disease-causing mutation, but also overall some genetic background and possibly genetic modifiers. Clinical manifestation, neuropathology if you have, environmental exposure, and also how do you derive those primary cell cultures. So what we've started, and we have now 5 lines of those, because we're interested in the neuropathology matching with our donors. We were collecting meninges from our brain donors with confirmed Lewy body pathology. So this is what you see. This is a piece of meninges, and then these are the meninges cells that are growing out of this tissue. This is 17 days later, and we can bank those cells, and they grow very nicely attached in the tissue. The other area where you can see a lot of variability is the nuclear reprogramming process. And it's both very exciting to see that this method is working, but it also gets quite complicated. So you can use integrating methods, you can use non-integrating methods. And originally, when we derived our first lines, we used the Yamanaka factors and a lentivirus system. And since we do not want to have any integration in the genome, we are now using Sendai virus as a non-integrating method. But there are all other different forms around. People use mRNA, people use mini-circle DNA. Protein is also a way to make iPS lines. So it's really up to the user which system to use. But I guess the take-home message is that you want to be consistent. In terms of neuronal differentiation, there are also lots of options. So originally, probably 15 years ago, people used spontaneous differentiation of stem cells to derive neurons. Then in the early 2000s, there was a co-culture with stromal feeder layers called SDIA, stromal derived inducing activity. You can co-culture your cells with astrocytes. There's an ambroid body differentiation or neosphere generation methods out there. You can use differentiation using small molecule inhibitors or recombinant proteins. Or you can also engineer your cell lines to express certain midbrain factors to differentiate and push those cells towards the dopaminergic lineage. So I think most of the labs that I've been working with are now using what's here under number 5, so the differentiation using small molecule inhibitors or recombinant proteins. And this is just an example of how you can derive very nice dopaminergic neurons. You see here under D, these cells are stained with TH. And they go through a phase of low progenitors where they express really high level and keep high levels of FOXA2 expression, which is this red line up here under panel A. In general, I wanted to note that there is also variability in general culture conditions, so you should take a look at your seeding densities, passage number, how you passage your cells, if you do it manually or enzymatically. What media you use can make a huge difference. The extracellular matrix you're introducing, cryopreservation, and even plasticware, you should make sure that that's consistent and you don't have many changes there, which can affect the culture and the quality of the culture. Okay, in summary, the challenges and the solution that we have here is you should take a look at systematic collection and documentation of your patients. You want to collect matching controls. You want to genetically characterize your subjects and patients, and if you have a chance, you might also want to generate genetically engineered isogenic lines. Then In terms of iPS derivation, you want to use consistent methods. You want to use consistent consumables. And with every cell line, you want to make sure that your karyotype is not changing and you characterize your cells properly. Again, here for the neuronal differentiation, consistent methods are key. And also, you want to have an unbiased characterization and measures of your cell type of interest, for example, the number of dopaminergic neurons and the midbrain specificity. You also want to make sure that cells, if possible, are physiologically active. Okay, so I'm showing you here a very interesting study that we did a few years ago with two other groups, actually three other groups. It's Sangamo Biosciences. It's the group around Tim Greenemeyer and Ole Isaacson. And so we were using gene editing in patient-derived induced pluripotent stem cells to look at a specific phenotype that's caused by LRRK2 mutations. So what we first saw is in this assay that Laurie Sanders developed is that we have mitochondrial DNA damage in cells that carry LRRK2 mutations. And it's a PCR-based assay, and in this assay, the polymerase that she's been using would not amplify a template as quickly when there is mitochondrial DNA damage compared to a control set. So what you have here is you have on the x-axis the different clones that we've used, and then on the y-axis the number of lesions per 10 kilobase that she measured. So C123 are control clones, so there's very little mitochondrial DNA damage. But then when we're going into the iPS-derived neurons from patients with LRRK2 mutations, L123 with a G2019S mutation, you see an increased amount of DNA lesions, which is also true for this second LRRK2 mutation, R1441C. We compared the mitochondrial DNA copy number, but those were definitely similar across the line. So that was not the reason that we had fewer mitochondrial copies in the cells that we've seen the differences that are here described in panel A. So then we said, okay, it's an interesting phenomenon, but is this really due to this one mutation that we think of? And so we used zinc finger nuclease at that time, and nowadays I also want to use CRISPR, which is a little easier to design and also a lot more economical. But the concept is the same. So basically, the zinc finger nuclease is like a molecular scissor, and you can create double-strand cuts, and then depending if you have a homology donor, you can introduce homology-directed repair and actually correct a mutation, or if you don't have a donor, the cell will repair the break itself and through non-homologous end joining, but that process is prone to error, and in some cases you might introduce smaller deletions or also insertions. So that's a way to create knockout models, for example. And what I'll show you here is this is the design. So this is the zinc finger design that you see here. And the donor that we introduced had, interestingly, a silent mutation that we could use as a restriction site to genotype our sample. So that was quite an elegant approach that Cellular Biosciences developed at the time. So this is the results of our gene editing in these cells, and what you see in the table is the modification where we received gene correction, in this case on both alleles, Some of the clones that we characterized had monoallelic, non-homologous end joining on the wild-type allele. Some had non-homologous end joining on the mutant allele. And then for some, we couldn't quite figure out on which allele it was, so it's unknown. We also had biallelic, non-homologous end joining, so we created some lines that are actually a biallelic knockout, which are also quite interesting. And then in the system also, we don't always modify all of the lines, so some stayed unmodified, and for some we didn't characterize all of the clones. Okay, so here's now the experiment again that I showed you on the mitochondrial DNA damage, and in this case we compared the LARC2 mutant cell line, which is here, to now the corrected LARC2 line, which is now wild type. And on panel A and B up here, we did this assay, this PCR assay in cells derived from neuroprogenitors that are Nestin and SOX1 positive. And down here, we've used differentiated neurons that have a portion of pH neurons, but also beta-3 tubulin neurons. So it's not a complete dopaminergic culture, but we've also seen that we get an increase in mitochondrial DNA lesions in the mutant line, whereas when you correct with the gene engineering approach, you see basically no mitochondrial DNA damage anymore. What we've also done is we've repeated those experiments in a separate lab with a little different methodology. So we worked with Ole Isaacson and Oliver Cooper at that time, and Oliver repeated those experiments and were able to replicate this as well. That was a nice confirmation that this is a real assay and a real phenotype. So overall, we have in LARC2, G2019S mutations, mitochondrial impairment. And I haven't shown you all of the data. We have some additional assays that show that. We've seen that LARC2 can cause an increase in mitochondrial damage. And with zinc finger modification, we were able to reverse that mitochondrial damage. So in this case, we were able to consistently replicate these results. So we have different donors, as you've seen in the first few slides. We've used different iPSC clones and got similar results. We used, in this case, different neuronal differentiation protocols, replicated the results, and then ultimately we were able to confirm that phenotype using gene correction. So that was a very nice confirmation and validation that iPS modeling actually helps us understand Parkinson's disease. Now I'm coming to the end of my presentation, and if you're interested in this and want to share this, we have a blog that shows how to take a fibroblast from a patient. In this case, it's Alex, who had very early onset disease. He was 36 when he was diagnosed. He donated his cells, and he is in a dialogue with us, and he learns how his stem cells get turned into stem cells, and then how we differentiate them. So visit our blog. You can also visit us at the Cellular Lab on Twitter. And I want to thank everyone who has participated in these studies. First of all, I want to thank all the patients and volunteers that are putting skin in the game. I want to thank the former lab members who helped setting up the lab, building the iPS bank, and doing the early studies with me. My current lab members are listed here: Adrian, Faria, Farah, Jimmy, and Kelsey, and I know they're watching next door. I'm very thankful for being part of the clinical center at the Parkinson's Institute, and Dr. Livingston and Dr. Caroline Barlow give me a lot of support, as are our clinicians in the clinic. And then on the right side here, I have highlighted the folks at Sangamo, Pittsburgh University, and McLean Hospital, Harvard University, who helped with the IPS zinc finger model. Funding and support came early on from the Bloom Foundation and Unity Walk as seed funds. The California Institute gave us a first big grant to jumpstart the IPS bank, and we also have the Parkinson's Alliance, who helped us sponsor and fund this research. That's all. Thank you all for listening, and I'm ready for questions. Okay, so now I guess I'm reading the questions myself. Okay. Have you been able to see a phenotype in neurons that have been derived from iPSCs from both sporadic, from both sporadic and familial forms? So that's a very good question, because sporadic forms, as I showed you, don't have the typical mutations that are described in Mendelian forms. We personally have not delved too much into sporadic Parkinson's disease, as it seems a little bit more complicated due to the variation. But there are some papers out there that found common phenotypes between familial and sporadic forms. Okay, the next question here is: Have you looked at functional endpoints to assess mitochondrial activity in mutant lines? And then, I'm sorry, it looks like the lost. Oh, I see. Down here, I have to see. I look at them. Okay. Have you looked at functional endpoints to assess mitochondrial activity in mutant lines? Do you see differences between control and patient lines? Yeah. So we've looked at mitochondrial activity, and there's a nice paper that Adrian published in 2014 in PLOS ONE, where we were looking at changes, for example, in the mitochondrial transition pore, mitochondrial membrane potential, and we've seen differences. The earlier presented LARC2 triplication line. In that study, we've been using the neural precursors to evaluate mitochondrial function. Are you planning on expanding your studies to other patients with some other Parkinson's mutations? The answer is yes. I'm planning on that. The long list of mutations and fibroblast cell lines you see has some rare mutations that can also cause multisystem Lewy body disease. For example, the 22q11 deletion. We've just recently followed a patient who has an ataxin 10 expansion, which has never been associated with typical Parkinson's disease. Unfortunately, here we don't have neuropathology yet. So the next question, could this correction be applied to animal models? So I believe you're referring to the correction of the LARC2 G2019S mutation. I believe so, and zinc finger technology and other gene editing technologies are now used also to create rodent models. So I'm pretty sure that could be applied. Okay, next question here. You described a diverse but specific population of cells that are affected by Lewy bodies and Parkinson's disease. What do these cells have in common that renders them vulnerable to the genetic lesion? That's a really good question. And I probably don't have the answer here. My thought for the preferable vulnerability in dopaminergic neurons in the substantia nigra is that they project to the striatum and they have a lot of connections in the striatum. So one dopamine neuron can connect to up to 75,000 neurons in the striatum. On the other hand, there's also a lot of redundancy. So about 100 to 200 neurons in the striatum can be overlapping and innervated. So I don't know this answer, but it's a really good question. So, I mean, one way would be to either differentiate not only dopaminergic neurons, but also noradrenergic or serotonergic neurons from iPS lines. Or the opposite you could do is you could generate a more generic neuron in a dish and then see what's common. So there might be two other ways to do that, but it's a very interesting question. Okay, so looks like I've answered all of the questions. Thank you so much, guys, for.
Great, thank you, Bridget. That was a fantastic presentation and great job answering the questions and great questions from the audience too. If there are no more questions, I would like once again to thank Dr. Shuo for such a fantastic presentation. Do you have any final comments for us before we part?
I'm quite excited about this type of education and training. I mean, not everyone can attend conferences, and this is a way to learn about new science. I've myself signed up for another 2 Lab Roots webinars, so I'm quite excited about this format. Although it's a little hard to connect with the audience, but I guess you need to get used to that. Thank you so much.
Well, you did a fantastic job. And thank you again to the audience for joining us today and for some really interesting questions. We would also like to thank our sponsor, Cellular Dynamics, for bringing today's educational webinar to you guys. This webcast can be viewed on demand through September 2016. LabRoots will alert you via email when it's ready for replay, and we really encourage you to share that email with your colleagues who may have missed today's live event. Until next time, bye-bye.